Okay, so I would like to start out thanking Rachel for inviting me to speak here. It's a real honor. I'm really excited, particularly because it's a topic. I'm in the midst of finishing up a book for the lay public on the very same topic. So this is kind of a nice distraction from spending 10 hours a day writing on this book. And I also would compliment Rachel and her team for organizing this phenomenal event, and which is reflected in the amazing turnout. It's too bad that Sarkis, Mazmanian friend and colleague, has canceled in the last minute, because I was relying on him. He's a card-carrying microbiologist, immunologist, to really set the stage for the microbiome and the interactions with the immune system. And so obviously I cannot fill in for this. I added a couple of slides, but so you will not have the pleasure to get this wonderful introduction that Sarkis gives. Here are the four areas that I would like to cover in this talk, and I was told I could go a little bit over my assigned time. Just give a few background pieces of information about the magnitude of the problem of obesity. Highlight the two dimensions of food intake, which I think are important to also understand the neurobiological underpinnings of these two aspects, and which one we think sort of plays a bigger role in the current obesity epidemic. That will lead into this topic of food addiction, basically that one of these food intake components, the hedonic aspect, sort of gone out of control. And then in the last third, ask this question and give some answers, careful answers. What do the gut microbes have to do with it? So, and that question is not a question about what do they have to do with obesity, because we know that they play a big role in this. This is not my topic. I will sort of give a few pieces of evidence that the brain-gut axis with the microbes on one end also contributes to this to these altered ingestive behaviors. So one of the questions is, why should we care about the obesity epidemic? Everybody has seen this slide in many variations. So this increasing prevalence of obesity within the US, really focusing initially and continue to focus on the Southeast, but also affecting most of the other states. It is a fairly serious problem. If you look at the numbers, 2/3 of adults and nearly 1/3 of children struggle with overweight and obesity. The predictions, the projections are that this will continue, that approximately half of the population will be obese by 2030, even though I would always be careful with predictions. A lot of things could happen as so-called black swans, and these projections could change. And the burden on the healthcare cost is also phenomenal. And this is shown here. So in orange, you can see what the projected healthcare costs are. And then with different interventions shown with blue and the red lines, how this could be dramatically affected. So interventions that change behavior and public awareness could have a dramatic effect not only on the individual health and well-being, but also on the cost on the healthcare system. This is in some ways a worldwide problem with some of the highest rates in the US and Mexico, but also other, as you can see, other countries. It's hard to really extract any kind of meaningful difference. You could say, well, the lower rates in Italy could have to do with the Mediterranean diet, but that's really, we don't know that. So one thing has become very clear from the times when there was initial enthusiasm of molecular biologists that we could pin it down to one excessive secretion of a hormone like leptin that or the absence of this, that this is basically the explanation for obesity. So a lot of the initial research, the biomedical research, was really focused on a very reductionistic model that hypothalamus with its receptors for certain hormones would explain the whole story. This is certainly not the case, and we know now that many factors, some of which are not really directly related to the nutrients itself, but I'm just highlighting some. Anyways, so one of the areas that I'll spend some time on is this whole concept of the reward circuitry, what drives us to do things, and what role this plays. It's also related to the easy access to food, but also to the advertising industry, which, when we talk about in neurobiological terms, really targets the brain and not so much the gut, because all these visual and other sensory stimuli that we are given on TV and in the media, really all targeted at our centers within the brain that makes us crave what's being marketed. So many dimensions, certainly, and this is sort of the theme of the talk. One related to the food itself, the caloric density, the meal size, the easy availability, access, cheap food. But on the other hand, things that are not directly related to food, such as its physical appearance, smell, the way it's advertised, which targets a different part of the brain-gut axis. So a good question, or a very important question, is why do we eat? So do we eat? Clearly, all living creatures eat because they want to stay alive and they want to feed their metabolism. On the other hand, as humans, do we eat to satisfy hunger or to satisfy pleasure? You can see on top, these would be two meals that would be mainly targeted at the metabolic needs of the body. On the lower panel, you see two foods that really would attract mainly the interest and the motivation and the craving for a pleasurable sensation. Now, can we translate these two aspects into a neurobiological model that has to do with the brain-gut axis? And yes, we can. So I'm taking a couple of slides, modified slides from Nora Volkow's superb publications. She's clearly been the one person who has been pushing the concept of the reward aspect and the food addiction aspect of obesity single-handedly, and has made it sort of a key component now of this field. So if you look at the metabolic component of food intake, so we have several systems shown at the bottom from the stomach, which produces a hormone when the stomach is empty that drives us to eat, ghrelin. Ghrelin, interestingly, has a lot of effects, but it also can target the dopamine system and release dopamine. So probably one of the reasons that an empty stomach makes us want to eat something is because our dopamine system is stimulated by this hormone. There's a lot of other hormones, these so-called gut hormones that have been, so when I started my career at UCLA at the Cure Institute, at the Cure Center, this was the main focus of research. Strangely enough, at the time, these hormones at the time, nobody really knew exactly what they're good for. It was an isolation of one after the other. Every time a new one came, there was a lot of excitement. But at the end, this field kind of calmed down because people didn't really know why they are so important. So in the meantime, we know that the GI tract is the biggest endocrine organ of the body, secretes up to 20 different hormones in different endocrine cells. And the main effect of these, other than the stomach, which is the motivator to eat, are all inhibitory. So they're meant to shut off our desire to eat. And we'll hear about this later a little bit. What could go wrong in that system that drives the overeating. There's also the fat cells that produce leptin, and there's also the gut microbes that we'll hear more later. So the main research has really focused on characterizing this system, this metabolic system, and focusing on the hypothalamus, some beautiful scientific work, all the subnuclei, receptor systems of the hypothalamus that are responsible for receiving these signals from the periphery and translating them into behavioral responses. So here's just a list of all these various, I won't get into the details, but these various gut hormones that have been isolated initially with archaic extraction procedures from tons of pig intestine. Nowadays, we don't need to go through this, but it's kind of interesting to see something that took about 15, 20 years before the real importance of this research became known. So if you come back to our evolving gut-brain axis, so on top of the hypothalamus, there's other regions, particularly this region that is also the core element of the reward circuitry, the ventral tegmental area, and the nucleus accumbens, with dopamine being the main transmitter in this system. Very complicated system, but it's the main driver of any motivation that in any of our behaviors. And then on top of it, also very important, we have the prefrontal cortex, which is a main inhibitory system that suppresses the otherwise unrestricted motivational drive. So we have an elaborate interaction of, at the bottom, the metabolic mechanisms that are simply based on getting enough nutrients into the system. And then we have the higher levels that interact with the lower ones that are mainly driving food-seeking behavior, and with our prefrontal cortex and our cultural and family-learned traditions exert its influence on these lower levels. So at each of these levels, alterations could result in an overeating. So, oops, sorry. So if we add the external environment to it, so we'll come back to this, what I showed you earlier. So environmental influences are listed here that have primarily the target our brain, our prefrontal cortex, and many of them are subliminal. We don't think about this when we watch some advertisement for a hamburger at the right time on television, that this activates the entire system that I show you here. And suppresses signals that would come up from the intestine. And then on the lower end, the environment obviously influences what goes on in our gut. Food-related factors, and I've included drugs and antibiotics to it because we now know that their effect on the microbiota will also feed into this very complex system. So what are the evidence? What's the evidence for an alteration in this reward circuitry within the brain that makes us eat more than that our metabolism, that our bodies really need? So the appetite response to the sight, smell of tasty food is stronger in overweight, obese individuals than in lean people. So these central mechanisms, and you can study them without any stimulus in the periphery, are upregulated. Similarly, the taste or smell of tasty snacks triggers overconsumption in obese. So food preferences are altered. So food preference partially related to taste receptors on your tongue, which are also modulated centrally, resulting in that obese subjects have a stronger preference for highly appetizing, high caloric foods than lean individuals. So we have a lot of evidence that that these central factors are altered in a profound way in the obese. Now, like in many areas of neuroscience, brain imaging has sort of provided the neurobiological counterpart to the psychological and psychophysiological insights that have been gained before. So we know now that from not a huge number of studies, but a growing number of studies, some of which performed in our center, that there's greater brain activation in the reward system in response to high caloric food pictures. So without any ingestion of food, if you just look at the picture of a hamburger or of a steak, that your reward system shows greater activity. that there are not only functional but also structural alterations. I'll show you one example for that from our own studies. That high fat and high sugar food activates the mesolimbic reward, gustatory and oral somatosensory brain regions. And another study has shown that sugar is more addictive than fat based on this technology. So from the group of Dr. Volkow at the NIH, so they used a so-called PET ligand, a PET tracer. So this is a ligand for a receptor, in this case the dopamine 2 receptor, that using a particular brain imaging technique, positron emission tomography, will give you a signal based on its the availability of binding sites. And what they showed is that that in obese individuals, so raclopride is the name of that tracer. You can see the images for, you see up there the basal ganglia with the nucleus accumbens for controls, and for obese individuals that the binding potential is lower in the obese individuals, so fewer receptors for the dopamine. And on the lower image, a PET perfusion image, so looking at blood flow with O15 water, and you can see there's a higher perfusion in these nucleus accumbens areas. So both functionally and structurally, we know that the area that has the dopamine receptors is altered in a way that it will need increased amounts of dopamine in order to get the same satisfaction. So there has to be an overstimulation of the system. So I'm taking together a lot of information, some of them listed here. This concept of food addiction was formulated in developed countries. So some of these reasons include that the majority of people no longer eat to survive, even though a significant number of people go hungry every day. So when I always read these introductions of papers, it said there's no, people don't eat for the metabolic need anymore. It's actually not true. There's significant percentages of the population, even in the wealthiest countries, that go hungry. We've heard about this already, that eating is controlled not just by these homeostatic mechanisms, but also by these hedonic. Then animal studies in the early 2000s showed changes in behavior that indicated that was consistent with the same effects of drugs or of drugs of abuse. And so based on a lot of data, both in preclinical and in animal studies and in these dopamine and brain imaging studies, this concept of food addiction, basically a dominance of the reward system over the homeostatic mechanisms, was formulated. And this is just summarized here again. You can see that both drugs of abuse and high caloric and highly appealing food would activate the same mechanisms all the way up to the prefrontal cortex, with the prefrontal cortex basically being insufficient to counteract the activation of the mesolimbic dopamine system. So to show you a short summary of a study that was done in our group with Dr. Gupta from our group, so leading this effort. So we used a technique called diffusion tensor imaging. This is another brain imaging technique where you can visualize white matter. And I'll show you an example of this in a minute, what this looks like. So the connections, the fiber connections between different parts of the brain. And we looked at a fairly large number of people, both males and females. And they included both lean on the one side and overweight and obese on the other. We combined the overweight and the obese. Just one word about these white matter connections, what that is. So it's basically like the freeway system in the brain that connects different parts of the city. And on the right side, you can see what an actual image of these, what kind of image you can obtain from the brains of living human beings. And this slide basically, so you see these fiber tracts, and the color coding is the density of these connections. So the number of fibers that connected. So by using a very sophisticated mathematical technique where you throw in all the data without, so was kind of a taboo only 10 years ago, but big data analysis is now kind of a accepted technique that you let the data speak for themselves and look for the patterns in your data to tell you what may be abnormal, so without a hypothesis. And so what we could show, what this graph shows, is that we could separate the obese from the lean simply based on the findings that the computer pulled out from a vast database of these white matter properties, the number of connections, direction of connections within the brain. And which is particularly nice in this, it shows you that the regions that showed altered connections include the very regions that we're concerned about with the altered reward system, so the basal ganglia and the nucleus accumbens and the striatum. So this blind technique took a lot of data from people and could simply, looking at the brain without knowing anything about their bodies or what they look like, tell who was in which of these groups, and they identified one of the main alterations in the brain that this algorithm worked was the alterations within the reward system. So the take-home message for this is that obese and overweight subjects display changes in the white matter connectivity, in the freeway system in their plane, as well as regional gray matter changes in areas involved in reward, somatosensory processing, and salience detection. So imagine you don't have to be obese. If you're just overweight, your brain is not the same. Your brain is structurally remodeled from the influences it gets from fat cells. So we think currently that cytokines and signaling molecules, so-called adipokines produced by this excessive visceral fat, signal to the brain and change the structure, remodel your brain. So next time you plan to go on a diet, actually it's worthwhile doing it because you have a major effect on your most vital organ, the brain. So so I told you a lot about the brain-gut axis, and there would be a lot more obviously in details. So the question is, what do the gut microbes have to do with it? So the simple answer is, it seems like in biomedicine in general, the gut microbes seem to have to do something with everything. There's virtually no field anymore that this fascination with looking at the microbiome, from chronic inflammatory diseases to neurological diseases, where this focus has not happened. And it's too bad that you couldn't get the introduction and the really impressive details and facts from Sarkis. But I mean, just to give you sort of a couple of, this has made it through the lay press in the meantime many times. So there's about 100 trillion of these microbes inside of us, 10 times more than we have human cells, because they're so small and invisible. We don't really, so the size of that forgotten organ would be about the size of the liver, but because the cells are much smaller, so it's much smaller. It's distributed from your small intestine all the way down to the colon, the highest densities being in the large intestine. What's even more interesting is that so it's not only a 10-fold increase or greater number in the number of organisms, but there's at least 100-fold difference in the number of genes that these organisms have. Fascinating story from evolution in the oceans. The biggest biomass in the oceans are not the whales, but the microbes live there. However, billions of years during evolution, they had time to exchange genes and basically produce many of the signaling molecules that our brain currently uses. So the fact that they have so many more genes is actually an indication they've been around much, much longer than humans or mammals have, and that they had a lot of time to play around with these genes and evolve. There's thousands of bacterial species that live in the human gut, and many divisions. 2 bacterial divisions predominate. 90% of are these Bacteroides and Firmicutes. And many initial studies have shown that the ratio between those 2 major divisions varies in different diseases. But that's really just saying, like, Bacteroides and Firmicutes are like between vegetables and meat when you go to the supermarket. So it's a very rough characterization of differences. So this field of the microbiome and obesity started several years ago with Jeff Gordon at Washington University, publishing a pivotal study in Science that the obesity phenotype could be transplanted from obese mice into lean mice simply by transplanting the microbiome. A lot of press at the time. Now, much more sophisticated studies from the same group in the meantime came out recently, just a year ago, where they colonized germ-free mice with stool samples with microbiota from twins that were they were discordant for their phenotype in terms of body weight, obese and lean twins. And they could actually transplant, so these were genetically identical twins, but they differed on their, so there must have been some other factors that explained their obesity. And they provided pretty good evidence that that factor was in the microbiome because they could transplant from the obese twins into a germ-free mouse, and that mouse became obese, and from a lean into a germ-free mouse, and that mouse did not change its phenotype. They could then also show, once they had transplanted these microbes into the mice, when they co-housed the mice, that under certain conditions, namely when the food supply was healthy for the mice compared to our human diet, that the obese mice adopted the lean mouse phenotype, so it became lean, just living. So if you eat healthy and if you marry a spouse that, if you're obese and marry a spouse that's lean, you have a good chance that actually you may lose weight as well. But that did not happen. That did not happen when the diet, when the caloric content, the fat content in the diet was increased. Then this transfer or this exchange of phenotype did not happen. So showing the interactions of, so the genetics didn't seem to be playing the major role. It would seem to be the microbes with the metabolites, and they showed what metabolites in these microbes produced. But also a very big factor was what they actually consumed. So we don't get away. So the simple therapy for obesity will not be the fecal transplant, which is now being done for tried for many disorders. But you still have to eat the healthy food, otherwise it's not going to work. So I'm going to skip the details of this. So the microbes, the microbiome in the brain-gut axis that we knew already a lot about before really happened within the last 5 to 7 years, so a very new field. A lot of very exciting data. In animals, you seem to be able to influence just about anything from eating behavior to mood, affect, social behavior. We don't know yet. Unfortunately, there's a real scarcity of human studies. How much of this almost futuristic kind of science will translate ultimately to humans? But this is kind of the picture that has evolved from these animal studies that with multiple pathways that connect the gut, shown on the lower side, with the brain, and I've shown both the rat brain and the human brain. We'll say in a minute why that is. So many of the metabolites that about 5,000 metabolites that these microbes produce can either go into the bloodstream and act like a hormone going to the brain, and if they pass the blood-brain barrier, then they may have an influence directly on the brain. Or there's other mechanisms. They could act on vagal sensory fibers, both in the gut, also in the liver, and then influence the signal that goes into our homeostatic control centers in the brainstem and into our limbic system, and thereby modulating brain function. So a complex system. We know something about this now from animal studies, and we know almost nothing from human studies. So just give you one example. So there's a lot of studies, and it's very well supported, similar to what I showed you earlier from Jeff Gordon about the role of the microbiome and its metabolites in obesity. Many of these things do not require the brain, or nobody has looked at it because many of these metabolites go directly to the liver, change metabolism, influence the metabolic development of metabolic syndrome. Now, there's a couple of studies that I was going to mention. One is, this was a study that were, so they used these toll-like receptor 5 knockout mice, which have an altered immunity, and also develop an altered microbiome. So you do something on these mice that changes innate immunity mechanisms, very profound mechanisms. These mice have a different microbiome. They develop the metabolic syndrome with many of its features. But interesting for this discussion is they also become hyperphagic, so they eat more. Most interesting, they take the stool of these mice, transplant them into the mice that have grown up without any germs, and now the germ-free mice will become hyperphagic. So somehow you have transplanted a signal that goes from the microbes to the brain that makes these mice eat more regardless of the food supply that they get. So nobody has followed this up, surprisingly. This field of microbiome, as I mentioned, gut-brain interaction is new. There's only a handful of us who sort of circle the country like on a road show, because there's so few of us, so we always speak together, including Sarkis. But the potential, as you can see from something like this, that something in our microbes makes us eat more, is kind of intriguing. And somebody has published a very speculative, purely speculative review article on this, which received a lot of attention in the lay press, with the title, "Is Eating Behavior Manipulated by the Gastrointestinal Microbiota?" So this is basically a concept that these microbes produce substances, metabolites and hormones, and neuroactive substances that somehow have access to our dopamine system and play a role in driving food-seeking behavior. In this article, they suggested that basically the microbes are the much smarter part of our organism. They have a lot more genes. They've been around billions of years. They figured out ways and that can make us eat what's best for them. So it's a fascinating theory. I think we'll see in the future if that really is the case. So more realistically, how do we study these gut microbiome brain interactions? So there's several mechanisms shown on the lower part of the slide, and several readouts. The main difference is that in animals we have to look at behaviors. In humans we can use brain imaging to see structural and functional changes, and we can look at subjective reports. So this is what has come out of some 40 studies that have been published in the last several years on this topic, that that the gut microbiome plays a major role in nociception. And you can see that, I've listed here not the human words, but it's either measured in reflexes or behaviors, or emotional behavior, social behavior, ingestive behavior. And at the brain level, that any manipulation of the gut microbes results in profound changes in the biochemistry of signaling molecules within the brain. So many people have speculated this, so there's got to be a human counterpart to this. We did one study, and I'll show you just a couple of things on that study, in healthy people, where we tried to manipulate the gut microbial either composition or their function, their metabolites that they produce, with an intervention, the intake of a probiotic mix, a cocktail of several probiotics, over 4 weeks, and we looked at them behaviorally and with brain imaging at the beginning and at the end of that period, shown here. And just so to show the one highlight of that study is that we basically compared 3 conditions in that study. One were these subjects, and I should emphasize, healthy, young, completely asymptomatic women without any elevated anxiety or depression symptoms. So the ones that took the probiotic cocktail. We could monitor this on their stool analysis that they actually took the probiotic. Was one group. The second group was they got a non-fermented milk product without any probiotics in it, and the third one didn't get anything. Then we asked on our computer analysis what network of regions in the brain changed from the beginning to the end of this intervention, and was there a difference in the way these regions were connected with each other. And what we found is, to our surprise, really a fairly extensive network in the brain containing sensory, visceral sensory regions that became less connected in the probiotic group, showed no change in the placebo group, and actually increased in their connectivity in the no treatment group. Now, what triggered that network activation? It was a task often used in psychological and psychiatric research, this face recognition task where people have to look at these images that are shown on the left side and had to match the two that had the same facial expression emotionally. Very ancient reflex, very ancient emotional reflex. So the way the brain processes that reflex was altered, to our amazement, by basically just ingesting a mix of microorganisms, beneficial microorganisms, over time. What that means since we didn't have a patient population, we don't know if that would decrease anxiety, if it would have any behavioral correlates. So just to summarize this again, and I should mention in that study, the composition of the microbes did not change, but the metabolites that they produced, your gut microbiome, with the probiotics, the metabolites that they produced and released was different, and this is just one more summary of this. The microbes are a factory of metabolites, thousands of molecules. 40% of all metabolites in our circulation are gut-derived. So who we are and what we do and how we think is probably to a major degree influenced by this, and we have no idea at the moment which one of these metabolites, alone or in combination, patterns of them produce what kind of central changes. These metabolites, some of them go through the liver, some go directly to the brain or to other organs, and they basically have an effect on all the organ functions. Best studied in the liver for metabolism, metabolic syndrome, but now increasingly studied also with their effects on the brain. For example, Sarkis would have presented this example. They recently identified one single metabolite that was increased in an autism-like model, in an autism-like mouse model, and that they could take stool from autistic children, transplant them into germ-free mice, and get the same social interaction alteration in these mice related to this one single metabolite. So you can see that this has the potential of really having major influences on medicine. We also did this study in a group, and I'm not going to get into detail, just illustrating this, that these metabolites are indeed related to the brain, to brain structure. We identified a set of metabolites in this study where we looked at healthy control subjects and patients with irritable bowel syndrome, identified what metabolites differed between the two groups, and then those that differed did a correlation with brain structures in this machine learning approach. And there were brain structures directly correlated with these altered metabolites. I'm almost at the end. Let me end with something. So Eric Dodson, one of our collaborators, he's a surgeon who doesn't really agree with that statement, but sadly, despite all the things that we've learned about obesity and we're learning about it, the only thing so far that works is surgery. And it's pretty dramatic with some exceptions. So some people do not respond to it. We don't know why. But the reason I bring this in because that points potentially also to a major role of the microbes. It's different kinds of these bypass, so-called bariatric surgeries. Initially, it was thought, and these surgeries were developed that this is a mechanical, either a obstruction, the flow of food into the small intestine would slow down your eating behavior, or there would be malabsorption. So many of these mechanical, simple mechanistics do not really explain the full spectrum of what happens during these surgeries. And I just want to draw attention, so this is a summary of these various effects that could play a role. Just want to draw attention to the brain part of this. There are some immediate effects on appetite, on food intake, on food preferences. So when you undergo the surgery, your preference, what you like to eat, is changed. And it's pretty rapid, and it's quite possible now that one of the major effects that these surgeries have is on the central control, counteracting, for some reason, this overactive reward system. And one of the questions is, could the microbes have something to do with it? So the microbes are profoundly altered in these surgeries because you have different transit rates and secretion. And some experiments in animals have shown now that not only are the microbes altered if you do this surgery in a mouse and a rat, but if you then transplant to the standard technique that you're now familiar with, you take the microbiota of these surgically treated animals that have lost weight and put them into a normal germ-free mouse, that mouse will also start losing weight now. So again, something profoundly changes in the microbiota that has an effect on central systems that drive food intake. So I'm going to end with this slide, basically just taking a schematic that Nora Volkow has proposed. So you heard a lot about this homeostatic, hypothalamic-driven regulation of food intake that's extremely well studied in humans. The newer dimension of the food addiction, the cortically and reward system-driven ingestive behavior. And what I've inserted here is the gut microbiota, which seem to be able to influence, in theory, both of those systems in a profound way. So if this will lead in the near future to these dramatic changes on the healthcare system is another question. In the meantime, I want to thank the people that have generated these data in our group. Annie Gupta, whom I could not find the right picture this morning, is not included in this, so she has done some of the major studies on these correlations of structural brain changes with the obesity phenotype. So thanks for your attention.